Hello, you guys. This one is going to be on cells, a tour of the cell, chapter 6. Your must-knows for this chapter are the differences between prokaryotic and eukaryotic cells, structure and function of the organelles, the structures that are common to plants and the ones that are common to animals, and the organelles that are found only in plants and only in animal cells. So just a quick review of cell types. There are really two types. Prokaryotic cells are bacteria. They are small, very, very small. They are very simple structures, especially when you compare that to the eukaryotic cells. Protists, those are single-celled eukaryotic cells, fungi, plants, and animals. Here's a picture. The prokaryotic cell on the left, notice the scale, 0.5 micrometers. Whereas the one on the right, that's 10 micrometers in that scale. And so if you were to put that up against the cell, that's probably 20 and a half micrometers across or lengthwise compared to just this little 1 micrometer guy over on the left. So they are much smaller. Prokaryotic cells are much smaller than eukaryotic. They also have very simple structures. They are just membranes. Usually they have some kind of projection. This shows you an example of fimbriae and flagella, two different types of extensions used for mobility or moving around. They have a nucleoid region where their chromosome is kept. They have a long chromosome, but it is one long continuous piece. It doesn't have a bunch of strands like eukaryotic cells. They have a cell wall, many of them do. That is a structure made out of proteins called peptidoglycan. They have ribosomes, which they use to build all of their proteins that they need to make and use energy. So if you were to go back through and just know the difference, prokaryote, just name only, is before a kernel. Kary means kernel. Kind of a weird term, but if you think of like a kernel of corn, it's basically a small little round piece on a bigger piece. So this is before there were these little smaller membrane bound internal pieces within cells. They evolved first, whereas eukaryote translates in Latin to eu, meaning true, and kary, meaning kernel. So this is a true kernel, a true little packet inside the cell. So that's ended up being a nucleus. This kernel that they were looking at and observing was the nucleus. Prokaryotes do not have one. Their DNA is found in a nucleoid region, which is just a spot in the cell that doesn't have as much cytoplasm in it so that the chromosome has space to exist. Versus a eukaryote has a nucleus and a defined nuclear envelope separating it from the remaining cytosol or cytoplasm. Both of them have that. There are no other organelles in a prokaryote besides its ribosomes, which are not really organelles. We call them that sometimes, but they're basically little packets of RNA, which are just all lumped up on themselves. So they have DNA and RNA, but not really any other membrane-bound internal structures. Whereas eukaryotes have a whole bunch of specialized membrane-bound organelles. Prokaryotes are small, eukaryotes are large in comparison. Prokaryotes are very primitive, meaning they're very ancient, and evolution has kept them mostly the same. They have not changed too much. Whereas eukaryotes have changed a lot over time and evolved to be much more complex. Examples of prokaryotic cells are bacteria and archaea. And archaea are just really, really ancient prokaryotes that live in extreme environments and do osmosis and chemisynthesis of all kinds of different compounds. They kind of don't really fit in any category, so we gave them their own. And then plant and animal cells are the most common eukaryotes. Here's some fun interactives for you guys to do. I recommend the one, I think my favorite one is the one on the bottom, Scale of the Universe. These links are clickable within the PowerPoint on the website. So please check them out and click through them. They're kind of fun. Cells have to be small, so cell size. In order to maintain a large surface area to volume ratio, cells must be small. Now, why do they have to have a large surface area? That allows an increase in rate of chemical exchange between the cell and the environment. In other words, the materials that they rely on to stay alive and to keep making their proteins have to diffuse passively into the cell. So if we're relying on this random diffusion, they have to remain small. Otherwise, the diffusion alone will not account for the entire volume of the cell inside. Here's a picture from the textbook that actually is kind of complicated. It looks simple enough, but being able to use the diagram to explain why cells are small is a good skill to have. So what I want you to notice first about the diagram is you have this small little cube over there on the bottom left. So and it says 1 next to it. Just imagine 1 is just any unit. 1 centimeter or whatever. It's a cube with only 1 unit on every side. So its total surface area is 6. It has 6 sides, each measuring 1. Now, if that cell were to get larger, if it were to grow to be 5 times as large on all the sides, we now all of a sudden have a total surface area of 150, which sounds pretty big, right? But let's look at the blue row down there at the bottom. If each side is only 1 unit, that means its volume is just 1. But then if it gets bigger, its volume increases to 125, right? Just like its surface area gets big. But now if you look at the ratio between how much surface area is available for diffusion versus the volume of materials inside that have needs, we now get a reduction as the cell gets bigger. So if you look at that first column underneath the little cube that says 1, surface area 6, volume is 1, which means the surface area to volume ratio is 6. Now look at the large cell, that's 5, it's grown bigger. 150 is the surface area, so it's pretty big. The volume is 125, which is also really big, which means if you were to look at the ratio between the two, we're now down to only 1.2, which is not good. That is not enough surface area to account for all of that volume and all of the material and space within. So in order for a cell to, or in order for an organism to grow, you can't just let the cell get bigger. Instead, you have to add more cells. So that's what happens in that last column. It's showing that an organism can be large, but it's going to have to add a bunch of smaller cells. Keep the cells small, but have more of them, and that maintains a surface area to volume ratio conducive of diffusion. So that's where multicellularity comes from. These organisms, like us, are trillions of cells big. We are large, but it's because we're made up of many cells, not because our cells are big. That's what that diagram means. So an example of surface area in an animal is our intestinal tract is lined with a bunch of folded membranes, and then even those membrane folds, each cell has membrane folds on it. So it is a lot of surface area, which is good because think of the function of that organ. Your small intestine is there to absorb. The villi are what those folds are called. Microvilli are even the smaller folds on top of each individual cell. This is just a close-up of that. Root hairs on plants are another example of increasing surface area for absorption. So if you look really closely at a plant root, especially here a germinating pea, it has not only a root for absorption, but on it a bunch of tiny other little micro hairs in order to increase surface area and thereby getting all of its nutrients faster. Okay, here we go through the organelles. The nucleus, its function is called the control center, mostly because it contains the DNA. It is surrounded by a nuclear envelope, which is a double layer membrane, and it is double layer of phospholipids, just like the outer membrane. And it is continuous with the rough ER. So the endoplasmic reticulum is also a series of membranes that are coming off of the nucleus. The nucleus has holes in it called nuclear pores, and materials can enter and leave the nucleus through these holes. That's what you guys see on that picture on the right is a really, really close-up view of the nucleus, and you can see the tiny little holes. Within there is chromatin. Chromatin is loosely coiled DNA. It's DNA together with a bunch of proteins called histones, and they make up all of your genetic instructions. They organize themselves into chromosomes around those proteins. They can unwrap whenever the DNA is needed in order to make proteins, and they can coil and ravel back up whenever they are not needed. Also shoved in there is a nucleolus, which is a region where the RNA is getting formed, which is where it gets clumped together. This is a picture from the textbook that shows you a diagram of chromatin, which is the blue string. The histones are the purple spiky things. You can see a close-up of a nuclear pore over there on the left where the RNA ribosomes are about to leave, and they hop out onto the endoplasmic reticulum. Looking closer at those ribosomes, we call them organelles, but they are not membrane bound. They are simply composed of rRNA, ribosomal RNA, which is a nucleic acid, and some proteins that hold them together. They are made up of two parts, small and large, and there's a couple types. And this is new information for AP that is different than what you learned in regular. Free ribosomes, or the ones floating around in the cytoplasm, are different than the ones that are found bound to the ER. The free ribosomes are making proteins for use by that cell so that the cell gets its jobs done. And then the bound ribosomes are making proteins that are going to be exported out of the cell and used by the organism or put into the environment. This is because the ER is part of what's known as the endomembrane system. So as the ribosomes are building these proteins, putting these amino acids in their correct order, they are being folded and modified, but then they need to be packaged in order to be shipped. So the endomembrane system is how packaging occurs. Endoplasmic reticulum is a very fun long word. We abbreviate it the ER. That is a network of folded membranes and vesicles that are used to package and ship. The rough ER is just described by how it looks under a microscope. It is studded with ribosomes, so it looks like it has a rough texture. And the function of that portion of the system is to get those proteins folded correctly and get them packaged into vesicles so that they can go to the Golgi. The smooth ER does not have any ribosomes on the surface. It's mostly because the ribosomes have already been, they already left. They aren't on there anymore, but it's also there to function in a different way in synthesizing lipids from its lipid bilayer, metabolizing any carbs, breaking things down. It's also a good spot for detox. You have a lot of enzymes in there that are working hard to keep the cell clean and free of toxins. Here is the endomembrane system in a cartoon version. Picture and then a real version. You can see the rough ER looks more like a series of canals and channels, and the smooth ER is a little more, looks tube-like. The Golgi is also involved in packaging and shipping and getting things exported. The Golgi is a great spot for the proteins to put their final shapes on. So this is where they will take their final quaternary structure and get anything added to them that they may need in order to do their job outside of the cell. This is also where lysosomes are formed, which are other little vesicles floating around in the cell. We call this series of flattened stacks cisternae, which is divided up into two sides. There is the cis side, which is the side that is facing the nucleus and the ER, and then the trans side, which is the side facing the outside of the cell. So think of it as like you have packages coming in and then you have packages leaving. So the receiving end is going to be getting vesicles from the ER, and then the packaging or shipping side will be sending those vesicles out to the outer membrane or the membrane of the cell itself. So again, the cis face receives vesicles coming from the ER. The trans face is where they leave. So this is a dynamic structure. It has membrane being constantly added to it, and then membrane is being taken away as it is formed into vesicles. So it is constantly changing size and shape, and it is a very dynamic structure, which is why when you guys see it in drawings and you have to try to identify it, it can take all kinds of different shapes in drawings because it is a dynamic structure. It changes a lot. Lysosomes are made here on the Golgi as well. They function in digestion. Think of the prefix lyse, means to break or to split. This is where hydrolytic enzymes exist. You can get peroxisomes, which are a really cool special kind of lysosome that have hydrogen peroxide in them. Those are the ones that are going to explode all over the cell during apoptosis, which is basically programmed cell death. When a cell is too old to do its job, or it's making a protein that it shouldn't be, maybe because of an error in the DNA, it will destroy itself by breaking open all of these lysosomes. Lysosomes can also fuse with other vesicles in order to dissolve or digest things. And that's what you're seeing in the picture. An old mitochondria is no longer needed or useful, so it is being completely broken down using a lysosome and a little peroxisome in there that's going to digest it or break it apart. Vacuoles are kind of a generic term. They're little storage vesicles. They are membrane bound and they contain things like food, bits of molecules, water, minerals. You could have pigments in a vacuole. There's certain plants that are a color like a red onion has a bright color in it because it's storing pigments in those vacuoles. There are also things called contractile vacuoles that single-celled organisms rely on in order to balance their water. If too much water goes into the cell, they can contract this structure and it will squirt water out. That's something unique to plants. They're large central vacuole. Here's the endomembrane system review. I don't know why it's here now. Oh, because vacuoles. Okay. So coming off of the ER is those proteins, those amino acid chains, these polypeptides. They arrive at the cis face of the Golgi. They become folded, they get things added, they take their final quaternary structure, and they leave on the trans face as in a vesicle as either a vacuole, maybe a lysosome. That's coming off of the smooth ER. They're making those. And it's now got products in it ready for use. Some of them will then fuse with the outer membrane and its contents will be released into the extracellular space. Here's a review of an overall picture of an animal cell. Stuff you won't find in here, chloroplasts. You won't find a large vacuole. The vacuoles will be very small. You do not see an extra outer rigid structure called the cell wall. And then there's another structure called plasmodesmata, which we'll actually talk more about when we talk about the cell membrane, but it is channels or holes in cell walls in order to allow for the exchange of materials. And here is a picture of a plant cell with a large central vacuole, big green chloroplasts, an outer rigid structure, the cell wall, and holes in the cell wall. Things you do not see in this picture, lysosomes, those are only found in animal cells, centrioles, which are the structures that are involved in organizing cell reproduction and chromosome movement, and flagella. However, there has been some plant sperm or pollen that have used flagella-like structures for mobility. Looking at more organelles, the mitochondria is the site of cellular respiration. We're going to know much more about that in a couple weeks. They are double layer membranes. They have an outer and an inner membrane and folds of membrane in between called cristae. Lots and lots of membrane for lots of surface area for lots and lots of ATP production. Inside the cristae is something called the matrix, which is a fluid-filled compartment full of its own DNA. Mitochondria have their own DNA, separate from your DNA, found in the nucleus of your cells. It's a different piece called mitochondrial DNA. Chloroplasts also have their own DNA. They're very similar to mitochondria. They have lots and lots of internal membranes, folded stacks called grana, that are made up of membranous discs called thylakoids, and they are filled with a fluid called stroma. Well, they're filled with, and the inner space is called the stroma, and it's filled with fluid. And they contain pigments, the thylakoids do, called chlorophyll, and that is used for capturing sun's energy. We'll go into that in much more detail later. The fact that mitochondria and chloroplasts have their own DNA supports the endosymbiotic theory that they were at one point an individual cell, probably a prokaryotic cell capable of making its own energy, or in the case of a chloroplast, using the sun's energy in order to make their own food, and then were engulfed by another prokaryotic cell. This was a significant advantage in survival, and so that's why a lot of these cells exist. Some of the evidence for the endosymbiotic theory are the double layer membrane on these organisms. They have a membrane surrounding them, but they also have an extra layer of membrane, probably from being engulfed by others. And again, having their own ribosomes, their own genetic material, and their own mechanisms for replicating. They can replicate themselves inside our cells. You do not make mitochondria, they make themselves. And then you have, that was that last piece. They reproduce independently within the cell. Peroxisomes, again, a special type of lysosome that makes hydrogen peroxide. The reason, or part of the reason why you need the enzyme catalase is because this hydrogen peroxide could build up and destroy things. Cytoskeleton is the interior network of protein fibers that help give structure and support to the cell and provide a scaffolding for things within the cell to move around on. That consists of three main types of fibers: microtubules, microfilaments, and intermediate filaments. Microtubules are made out of a protein called tubulin, easy to remember because of the name and the way it sounds. They are the largest, thickest fibers within the cell, and so they provide shape and support, and they provide like a track for the movement of organelles along them. They form the spindle for mitosis. Whenever we talk about spindle fibers in regulating chromosome movement, those are made by the centrioles. That's microtubules. And they are also used as a component of cilia and flagella. Microfilaments are thinner and small, and they are made up of a protein called actin, and they support the cell on a much smaller scale. They also are involved in cell movement, but like on a smaller scale. Again, if you were a single cell, you might use these tiny little hairs in order to move materials within your cell to make arms, right? Like an amoeba does something called cytoplasmic streaming, where it puts all of its cytoplasm on one side of the cell, which causes a little bulge in the in the membrane, which acts kind of like a foot. It's called pseudopods or pseudopodia. Intermediate filaments are exactly what they sound like. They are intermediate in size. They're not big. They're not small. They are permanent fixtures. These are more like a skeleton for the cell, and they keep the organelles in place. The centrosomes are the regions from which these microtubules grow, where the spindle is made. It is made up of two structures side by side. The centrioles are within this structure called the centrosome, also known as a microtubule organizing center. They stay small until the cell is about to replicate or divide, and then all of a sudden the microtubules become large and are sent out to attach to chromosomes. Cilia and flagella have kind of cool structures that's shown in these pictures. Flagella are long, and there's usually not very many of them, and it's really useful for a cell to be able to propel itself, versus cilia, which are short, and there's lots of them, and those are more for being able to control movement in many directions. Flagella, it's usually just one direction. Like think of a sperm trying to get to an egg. It really only has to go one way. Cilia give the organism much more control. You also have cilia on the surface of your throat cells, your trachea, which is the wind tunnel to help keep debris from building up. And then females have cilia lining their fallopian tubes in order to keep the egg moving down towards the uterus as well. They have what's known as a 9+2 pattern of microtubules, which means that there is 9 little dimers or sets of 2 around the outside, and then there's these 2 microtubules on the inside that those fibers connect to, so that it has an interior structure, and all it has to do is constrict one side and it causes it to bend, kind of like a puppet. The extracellular matrix is outside of the cell, but it is still made up of cellular structures, glycoproteins, collagen, there's other fibronectin, there's all kinds of fun stuff floating around out there. And usually all that stuff is anchored to the cell itself by proteins called integrins, or any kind of integral protein. And then cell by cell, when the cells are side by side, these are called intracellular junctions. Animal cells have several different ways that they bind to other cells and make tissues. They can form what are called tight junctions, where the 2 cells are pretty much fused by the membrane to prevent any kind of liquid from getting through. Then there's desmosomes, which are kind of like rivets. They extend out, and then the other ones also extend out to hold or anchor cells together and fasten them together in large, strong sheets that can't be ripped or torn apart. And then there's gap junctions, which are just like openings that allow the free flow and share of materials between 2 neighboring cells. So that way, if one cell gets sugar, all the cells that are connected can share those materials. A little bit more specifics on plant cells. Oh, this is a long one. Is the cell wall. There to protect the plant and maintain its shape. It is made up of a starch called cellulose, lots and lots of sugars. And then just like gap junctions, unfortunately, a cell wall can block the free sharing or free flowing of material. So it has a hole in it that allows that material to pass through to the neighboring cells. So plasmodesmata are just like gap junctions, except in cell walls instead of cell membranes. Here's a quick review. Again, a really good idea to write charts like this down and organize your reading objectives in charts. Plant cells only, they're on the left, and animal cells, they're on the right. There's a video for you to watch that I encourage you to do so by looking at this PowerPoint again on the website, on my AP Bio website. So enjoy, and hope you guys, hopefully that helps you understand some of the more complex stuff about cells. Now I have to stop this. I just don't know how. Hang on.